Human Genome Project was an international scientific research project which got successfully completed in the year 2003 by sequencing the entire human genome of 3.3 billion base pairs. The HGP led to the growth of bioinformatics, which is a vast field of research. The successful sequencing of the human genome could solve the mystery of many disorders in humans and gave us a way to cope up with them. This plan was initiated in 1984 by the US government, and the project was formally launched in 1990. The Human Genome Project originally aimed to map the nucleotides contained in a human haploid reference genome. HGP states that genome of any given individual is unique. Human genome involves sequencing the genomes of a small number of individuals and then assembling these together to get a complete sequence for each chromosome. HGP still remains world's largest and greatest collaborative biological project. The finished human genome is thus a mosaic and not representing any one individual. Goals of HGP: to identify and map all the 20,000 to 25,000 genes in the human DNA from a physical and functional standpoint, to determine the sequence of the 3 billion chemical base pairs that make up the human DNA, to store that information in databases, and to discover more efficient technologies for data analysis. Allow the private sector to access the information and technology that arise from this project, to address ethical, legal and social issues. In 1990, the two major funding agencies, the US Department of Energy and National Institute of Health, developed an MOU in order to coordinate plans and set the clock for the initiation of the project. Most of the government-sponsored sequencing was performed in 20 universities and research centers like US, UK, Japan, France, Germany, Canada and China. A parallel project was conducted outside the government sponsorship by Celera Corporation, which was formally launched in 1998. These 6 scientists laid the backbone of Human Genome Project, and they are James Watson, Francis Collins, Robert Sinsheimer, Charles DeLacy, David Smith, and Jim Kent. In this project, two different and significant methods are typically used. One is express sequence tags and the other is sequence annotation. Some salient features of Human Genome Project are: our entire genome is made up of 3164.7 million base pairs. On average, a gene is made up of 3000 nucleotides. The function of more than 50% of the genes is yet to be discovered. Proteins are coded by less than 2% of the genome. Most of the genome is made up of repeated sequences which have no coding purposes. Area of achievements of Human Genome Projects are: genetic map, physical map, DNA sequence, capacity and cost of finished sequence, human sequence variation, gene identification, model organisms, and functional analysis. It started from September 1994 till 2002. Applications of Human Genome Project. As the goals of the Human Genome Project were achieved, it led to great advancement in research. Today, if any disease arises due to some alteration in a certain gene, then it could be traced and compared to the genome database that we already have. In this way, a more rational step could be taken to deal with the problem and can be fixed with more ease. These are some of its applications.